In this video, I will discuss the RAS inhibitors. Now, the RAS proteins historically have been very difficult proteins to block, and yet they're really important to cancer cells, and it's really important that we do manage to create RAS inhibitors. In explaining RAS inhibitors, I'll start here with the fact that all our cells have on their surface receptor proteins that help them respond to signals from their neighbours and from their environment and via various signals sent out by organs and tissues around the body. Some of those signals are in the form of little proteins known as the growth factors. And when the growth factors latch onto growth factor receptors on the cell surface, that triggers a chain reaction within the cell, and it culminates in the cell receiving the message to survive, but possibly also to grow and to multiply. And that's why this process has to be under tight control so that cells don't receive the signal to multiply when they shouldn't do. I'll go through this now in slow motion. Growth factor receptors then, they sit on the surface of our cells. There's lots of different types of them. Different cells have different growth factor receptors on their surface. When growth factors attach to growth factor receptors, those growth factor receptors change shape. Having changed shape, the two growth factor receptors can now pair up, and once they've paired up, they grab phosphates from ATP and they transfer those phosphates onto one another. That causes an internal change in that receptor protein, and now there are docking sites for other proteins that come in and attach to those phosphates. The docking proteins are the ones that trigger off these chain reactions within the cell that culminate in the production of proteins by the cell, which tell it to stay alive and possibly to grow and multiply. And if that signal is really strong, then the cell not only receives a signal to stay alive, it also receives a signal to grow, to multiply, to stay alive, come what may. The RAS proteins are part of one of the most powerful pathways triggered by paired up growth factor receptors. RAS proteins are also triggered and activated by other types of receptor on our cell surface. They're a central point of importance. They are a central way through which the cell is told grow, multiply, and survive. There are three main RAS proteins: KRAS, NRAS and HRAS, and as I've said already, it's been notoriously difficult to create drugs that block the faulty RAS proteins that we find in cancer cells. I'll say a bit more about why it's so difficult to block RAS proteins now. So RAS proteins are constantly tethered to the internal surface of the cell membrane, and they buzz around on that internal surface. And when receptors have paired up and they phosphorylate each other, docking proteins attach to those phosphates, and that docking protein attaches to a protein called SOS. So SOS is one of a group of proteins that attaches to those docking proteins that attach to the phosphorylated receptors. And SOS is a type of protein known as a guanine nucleotide exchange factor. And what SOS does is it tells RAS to let go of a little chemical compound called GDP and to swap it for a different chemical compound called GTP. As with ATP, the source of phosphate for the receptor, GTP also has phosphates that RAS can use. Once RAS has GTP, it now attracts in RAF proteins and it activates those RAF proteins. And the RAF proteins go off and activate other pathways and force those cells to grow and multiply and survive. After activating multiple pairs of RAF proteins, RAS switches back into an inactive state, and it does that by turning GTP, guanosine triphosphate, into guanosine diphosphate. And GDP no longer has the energy needed to keep RAS active. The mutated versions of RAS proteins that we find in cancer cells, they are either able to more quickly and more easily swap GDP for the more active GTP, or they are slower than normal at converting GTP back into GDP. One group of treatments we have been very good at developing over the years are kinase inhibitors. Kinases use ATP as their source of phosphate rather than the RAS proteins, which use GTP as their source of phosphate. It has turned out to be much easier to create kinase inhibitors that block kinase enzymes than to create RAS inhibitors. And that's because the kinase enzymes have a really deep groove in their surface where that ATP sits. There's room there to fit a drug, and sometimes ATP drops out and leaves the binding groove empty, and our drug can find its way in instead and block that kinase. With the RAS proteins, the situation is different. There's no useful groove or pocket in which a drug can fit, and also RAS has such high affinity for GTP that it's almost impossible to design a drug that could get in the way and could occupy that binding site instead. The binding site for GTP is almost never empty. Here I've listed some of the most common mutated forms of RAS proteins that we find in various different types of cancer cells. In bowel cancer, for example, you find the G12D version of KRAS, or you find the G12V version of KRAS. In melanoma skin cancer, it's more likely to be NRAS that's mutated. In head and neck squamous cell carcinoma, it's more likely to be HRAS that is mutated. And various different mutated forms found in various different cancer types. The one mutated form of RAS that we are now able to block is the G12C mutated form of the KRAS protein. Now that G12C version of KRAS is common in lung cancer cells, the most common version of KRAS in fact. And the KRAS G12C inhibitors that have been developed, they are chemicals that bond directly or connect directly with the abnormal C that's present, the abnormal cysteine present in the KRAS protein. So these drugs, because they need to interact with that cysteine in order to work, they're not going to be able to block any of the other mutated forms of RAS proteins we found in cancer cells because they don't have that cysteine present. But sotorasib and adagrasib, they connect with that cysteine, they block the G12C mutated version of the KRAS protein. And there are newer KRAS inhibitors being developed, more potent, more selective inhibitors, such as divarasib. But what about other mutations and other cancer types? And one cancer where we almost always find a RAS mutation is pancreatic cancer, but we very, very rarely find the G12C mutation in pancreatic cancer. But we'd love to have RAS inhibitors that we could offer to pancreatic cancer patients because those RAS mutations are so common, so powerful, we'd love to be able to block them. And there are now 3 new groups of RAS inhibitors being developed. We have some that block just one mutated form of one RAS protein, similar to the KRAS G12C inhibitors. We've got KRAS G12D inhibitors being developed. We have other treatments being developed that target all the mutated forms of KRAS found in cancer cells. And beyond that, we have some treatments being developed that will block any active form of any RAS protein. Here are just a few examples of the drugs being developed, the drug names down the left-hand side, and I'm going to pick out just one of them and explain it further, daraxonrasib. This drug is able to block any active version of a RAS protein, whether that's HRAS, KRAS, or NRAS. It is derived from a natural chemical compound that's been extracted from a bacteria, and that compound is called sangliferin A. Sangliferin A binds very tightly to a protein found in our cells called cyclophilin A. And what the developers have done is take just a piece of that sangliferin A chemical compound and attach it to a different little chemical structure to create daraxonrasib. And what daraxonrasib is able to do is it can still attach to cyclophilin A, but it actually distorts the cyclophilin A so that it now interacts with and blocks RAS proteins. So a little summary about RAS proteins and RAS inhibitors. KRAS is the most frequently mutated oncogene in human cancers. It has taken us over 40 years to find an effective way of targeting it. And now we're going to see so many of these drugs coming through in the years to come. Drugs that block just one mutated form of one RAS protein, drugs that block all mutated forms of one RAS proteins, and drugs like daraxonrasib, which block all active forms of all RAS proteins. They work through a variety of different mechanisms, and they are going to be vulnerable to drug resistance like our other targeted therapies are, but it does open up a whole raft of patients who could now potentially be given a targeted therapy.